Abdominal aortic aneurysm, or AAA, is not just a mechanical failure of the aorta, it is deeply influenced by the immune system. Various immune cells play a critical role in its development and progression, leading to the weakening of the aortic wall and eventual rupture. Neutrophils, the first immune cells to respond to injury, are key contributors in the early stages of AAA. These cells release destructive enzymes, such as matrix metalloproteinases, MMPs, which break down the structural proteins of the aortic wall. Additionally, neutrophils produce extracellular traps, known as nets, which further promote inflammation and drive smooth muscle cell death, accelerating the weakening of the arterial wall. Monocytes and macrophages are equally important. When monocytes are recruited to the site of inflammation, they differentiate into macrophages, which can either promote tissue destruction or contribute to repair, depending on their subtype. The inflammatory macrophages, or M1 macrophages, produce pro-inflammatory mediators like tumor necrosis factor, TNF, and interleukin-6, IL-6, which further degrade the aorta. In contrast, M2 macrophages, which appear later in AAA progression, secrete anti-inflammatory molecules, contributing to tissue repair and potentially slowing down the destructive process. Another critical player is the T cell family, particularly the CD4 plus T cells. These cells release cytokines that not only contribute to the immune response, but also directly influence vascular smooth muscle cell death, further weakening the aortic wall. Studies have shown that different T cell subtypes dominate at different stages of AAA, with T1 cells and their pro-inflammatory cytokines being prominent in larger aneurysms, and ITH2 cells playing a role in smaller aneurysms. Lymphocytes, including T and B cells, infiltrate the aneurysm and are associated with the degradation of the extracellular matrix. They, along with other immune cells like dendritic cells and mast cells, contribute to the complex inflammatory microenvironment within the aneurysm. What's particularly fascinating is the interplay between these various immune cells. Neutrophils and macrophages, for example, form a destructive cycle. Neutrophils recruit macrophages, and macrophages further activate neutrophils, intensifying the inflammatory process. As our understanding of these immune mechanisms deepens, it opens new possibilities for therapeutic strategies aimed at modulating the immune response to slow the progression of AAA. This video is based on the journal article The Role of Immune Mechanisms in Abdominal Aortic Aneurysm. Could it be a promising therapeutic strategy? By Rasit Dinc. The link to the article is provided in the description box below. Rasit Dinc has also authored several books that examine related topics within the realm of healthcare in the digital age. The links to these works can be found in the description box as well. If you found this information valuable, we encourage you to subscribe for more insightful content on the future of healthcare.